Okay, so firstly, good morning to everyone in the audience. I'm Minh, and today I'm going to present about the science of nanobots. So firstly, here are the table of contents and how you can have a brief look. And so firstly, we'll go through the definition. Nanobots are programmed molecules. So it is as small as the size of molecules, and it works on the micro to nanometer scale. And if compared, if the Earth is 1 meter, then the marble is the size of a nanobot. So now to the background. Firstly, in 1959, the physicist Richard Feynman, here he is, first mentioned the term nanobots. And in 1996, the scientist Robert Freitas wrote the first study of nanobots. And the problems before nanobots is the mistakes in disease and illnesses detection, and it could lead to incorrect treatment and worsen the disease and waste the money of the patients. But what nanobots bring is precise treatments in exact area in the body where humans can't reach, and will detect the illnesses perfectly. So now to the design. The nanobots are built by biological molecules, for instance DNA, with the molecular self-assembly process. So what is the molecular self-assembly process thing? It is the process where the DNAs or the biological cells were programmed, were coded to work on their own and build a nanobot. And with this process, the nanobots were coded and work perfectly. And the second one is they're built in laboratories. They're man-made by man-made. Okay, so now to the types of nanobots. So firstly, we have the nanobot vehicles, which is the non-biological nanobots. So firstly, the nanobots were have spiral-shaped iron made, the materials, and this is how they look. So the spiral-shaped nanobots can be coded to swim under the presence of a magnet, and their special double coating prevents the nanobots to hit into the dense tissues. And in the future, we will have drug delivery systems coated on the shell of the nanobot here to deliver drugs into the places where treatment is needed. So secondly, we have the biohybrid nanobots. So here are how nanobots can gene silence cancer cells. So firstly, we have the folded DNA barrel. So as you can see, the barrel is folded onto a lot of circle shape. And we have the endosomal breakup agent called siRNA, and with the DNA barrel and siRNA combined, it will lead to a typical design of DNA nanobot. And here's how it works. So firstly, you have the endocytosis process, which means that the DNA nanobot will move into the cell to form an endosome. And here we could have two processes. If the DNA nanobot is not suitable for silence the cancer cell's gene, gene silenced cancer cells, it will move out of the cell in a process called exocytosis. But if the condition is suitable for silencing the cells, it would release the endosomal breakup agent here. And now the third process is called the endosomal escape, and which is when the nanobot breaks the wall of the endosome and to the place where the cancer cells needed to silence. And here the last process is gene silencing, which would silence all the cancer cells and prevent the cells to be spread out and be a threat to the human body. So this is also the biohardware process. So here we have the sperm and a microstructure which scientists have developed, and these acts like arms for the sperm to swim, and it will move the sperm into the direct spot of the egg for the fertilization process. So this is the sperm nanobots, and as you can see, it has the microstructure, which looks like a 4-leaf clover, and it will direct the sperm into the exact spot where the egg is. So now we will head to how can we control nanobots. Firstly, we could control nanobots by magnets. So we need a microscope and an electromagnet, and it is controlled by a magnetic manipulation system. And with the magnetic fields, because the nanobots were mostly made by iron, then the electromagnet will charge the ends of the nanobots and will move the nanobots into the direction where it was. So the second process is by light. So firstly, when the light shines, the semiconducting nanowires, which is inside the nanobots, will get the light particles, and it will charge the semiconducting nanowires, and it will move the torque of the nanobots, which is like the tail of several cells, and it will move the torque and thus direction the nanobots to move anywhere the scientists want. So now I will get to the uses. Firstly, the nanobots were used to deliver the drugs and medicines to targeted and needed tissues to where the body needed it. And the second one is to gene delivering and replacing. Nowadays we heard a lot of genetic therapies, which is when the gene is damaged, and with the gene delivering and replacing, we could replace the gene with in the genetic therapy. And lastly, it would eliminate the cancer cells, as I've stated before. So what are the limitations and challenges of nanobots? So firstly, the uses of hazardous and materials and light could harm the scientists and the developers' body if not used carefully. The second one is miscontrolled nanobots. If the nanobots are not programmed correctly, it would like ran into the cells and cause a lot of damage inside of the body. And the third one is auto-replicating nanobots without stewardship of developers. If the developers didn't program it correctly, then the nanobots could replicate itself a lot of times and even like know what they are doing without the need of the coding process, and it could work on their own without the stewardship of the developers. Then it could develop strongly and could be a threat to humanity. Yes. So lastly, the challenges is the engineering challenge. So the developers have a challenge to build even smaller nanobots. So lastly, what nanobots hold in the future? Firstly, it will recover cellular injury. What is a cellular injury? That is when the cells are badly damaged and badly harmed by outside or inside processes. And with nanobots, they can recover and fix what causes the cells to be damaged that badly. And the second one is it will address and detect genetic issues. So it could detect why the genes are not working properly, and it could, with the fixing that I've talked about in the last slides, this could work out the issues and then fix and then replace a better gene. And the last one is it could help eradicate tumors. So because the tumors are one of the reasons to cancer, then if we help eradicate tumors, then the cancer cells, we could even prevent cancer in the first place. So to the conclusion, nanobots can bring a very bright future to us and to our health. And that is the end. Thank you for listening.